Nature, so there are details in that paper, and the data set and our computer code are also available in links in that paper. And so this talk, of course, is about cancer immunology. And here we show a framework of the cancer immunity cycle published actually by our colleagues, Daniel Chen and Ira Millman. And so in the cycle, first the tumors release antigens, and then these antigens get presented to the lymph nodes. And then activated T cells enter the peripheral blood. These T cells infiltrate the tumor and then kill the tumor cells when they recognize the antigens. So we have a general framework of how this works, but a lot of details, of course, are missing. And these details are very critical for our understanding of cancer immunology. So some of the questions, of course, that we could ask about that framework are, what are the different types of populations or T cells? Here on the previous slide, we just showed one type of T cell, but we know there are many types. And with single cell technology, we can understand that better. And then how are these different T cells distributed in the blood and the tumor and the normal adjacent tissue, the NAT? And what are their patterns? How do they flow from one to another? And which cells are active in each step? What is the relationship between the different T cell receptors and the antigens? And then how do they behave? How do they expand, in what compartments? And what is the relationship between these T cells and checkpoint inhibition therapy? In other words, therapy against PD-1 or PD-L1. And how is the relationship of exhausted T cells, which are presumed to be the targets of checkpoint inhibition therapy, is that important as well? And then can we use this data to predict which patients will respond best to cancer immunotherapy? So we used a technology, of course, which is very popular now, called single cell technology. And of course, the most prevalent type of single cell technology is called single cell RNA-seq. And in this one, we take each RNA transcript shown here in this box here, and we can analyze them. We can group the cells by their transcript profiles. But a relatively new technology is called single cell TCR-seq for T cell receptors. And in this case, we sequence the alpha and the beta chains of the TCR receptors. And then when we match the sequences, we can group cells together if they have the same alpha and beta sequences. So these what we call clonotypes. Each of these big black circles, ovals, is a different clonotype, and the cells are grouped by clonotypes. So we performed both single cell RNA-seq and single cell TCR-seq in a set of patients. And we applied it to the tumor, NAT, and blood from 14 patients. Actually, the blood only came from 4 of the patients. But in all the other patients, we had both blood and NAT samples. And this yielded a relatively large data set. In fact, I think this data set, certainly at the time of publication, was the largest data set of T cells from tumor tissue. And we, of course, with the single cell RNA-seq, you can see the numbers here, and the T cell TCR-seq, because of the difficulty in the sequencing, we can't always get a TCR sequence in every cell in the corresponding 2 libraries. But we did get about 57,000 distinct clonotypes in 100,000 different cells that had TCR sequence available. So what we first noticed was that there was extensive sharing of clones in tumor and NAT. So if you match up the clones of the cells and group them together, you find that we can classify each of these cells or clones as each of the clones as being one of these types. So either a clone just has a single cell, which we call a singleton. So if that single cell was in the tumor, we called it a tumor singleton. Sometimes you use the small letter T here. And then again, if we had only one cell in the normal adjacent tissue, we call that a NAT singleton. So I also use the color yellow and orange for these. But if it had multiple cells, it was either multiplets. If they're all in the tumor, we call it a tumor multiplets. If all the cells in that clone were in the NAT, we call that a NAT multiplet. And if this clone had cells from both tumor and NAT, we called it dual expanded. And when we plotted this for each of the patients, we saw that a large number of clones actually were dual expanded. Of course, they varied across patients, but you can see here in lung 2, 162 clones had cells in both tumor and NAT. Likewise, lung 4 had a lot. As I said, it varied. So in this patient, colon 2, we had 0 of the dual expanded clones. So, and so when you grouped all of these clones together by these bar plots, and again, using this color code here, oops, you can see that most of the clones were singletons. You can see by the large numbers here, 1385, 2474. So most clones did not expand. They were singletons. But about 15% of them did expand. And, and then some of these were dual expanded clones. But if you looked at the cell level, so of course, because the cells, because of this expansion, you get an increase. And then here I'm showing them separately for the NAT and for the tumor. So the cells in the NAT we're going to count and the cells in the tumor count. You see this large area of purple, which means a large fraction of cells actually came from the dual expanded clones, with some coming from the tumor expanded and some from the NAT expanded. But still, the predominant story was there's a large number of cells in the tumor and the NAT which came from dual expanded clones. And so, so that's an interesting finding that we found in our paper. So to understand this further, we plotted the data not just in terms of a Venn diagram, but we plotted it numerically. And when you plot it numerically, you get these plots where on each of these patients, I'm showing the fraction of the clones that were in the NAT compartment on the horizontal axis, and the fraction of, or this is actually the cell fraction, the tumor fraction of the tumors, the cells that were in the tumor cell fraction, and I'm plotting by the position where they're located in tumor NAT. And you can notice that if there was only one copy in NAT, you get yellow, one copy in tumor, you get orange, and this is corresponding to the NAT multiplets. This is corresponding, this pink part corresponding to the tumor multiplets, and the part of the original are the dual expanded clones. And you can see there's a fair number of dual expanded clones. But the interesting thing is that a lot of these dual expanded clones seem to lie near the diagonal. You can see this most strongly for colon 1. You get a little hint in some of these other patients, not perfectly along the diagonal, but there's enough here to suggest that there's some sort of parallel dual expansion going on, meaning that about this, these clones have about the same number of cells in both tumor and NAT. And so that raised an interesting hypothesis. Yes, question? No.
Yeah, apologies to interrupt you. I'm very sorry, but just, in order for me to be able to follow what you're saying, yes. When you're referring to expansion, does this refer to pre versus post treatment?
Oh, I see. No, it just simply means the number of cells in the clone at a snapshot in time. And this is all this data we have is pre-treatment.
Okay, thank you.
So, so we just simply look at the population at a snapshot of time and say, did this clone have one cell or does this clone have multiple cells? And so we called, if it had multiple cells, we called it expanded.
Okay, thank you. Sorry.
I will talk a little bit later about pre and post treatment. So, but for our data, it's all pre-treatment. So this raised the interesting hypothesis: how is that we get this behavior of about the same expansion of every clone in both tumor and NAT? And so we raised the hypothesis: could this be because they're coming from a common source, as is peripheral blood? So as I mentioned, we got samples from 4 of our patients in peripheral blood. And these are the patients shown here. 3 of them from renal, 1 was from lung. And here I'm showing exactly the same plot as before, with the position of the dot indicating whether or not it was the extent to which it was expanded into the compartments. And again, you can see this strong, striking lining up of positions along the diagonal, at least in some patients, but not all. And by the size of the dot, we're going to indicate the clone size. So the large dot means they're expanded heavily in the blood compartment as well. And so that's interesting because you can see that the dots along the diagonal are also large, supporting our hypothesis that the expansion in both tumor and NAT could be attributable to the expansion in the blood. And conversely, in the patients that didn't show this parallel dual expansion, we didn't see much expansion in the blood. So to quantify this further, we used an expanded Venn diagram. This is the same Venn diagram we saw before, but now we're going to add two more categories. This green oval to indicate that they were not expanded in blood, and this brown oval to indicate they were expanded in blood. And then, of course, this part between the ovals meant they didn't appear in blood at all. So we're going to use these color codes and these diagrams. And the first thing we said was, these four patients differ in the amount of peripheral clonal expansion. In other words, the total number of cells that were blood expanded versus non-blood expanded. You can see this graphically from this picture. The total number of large dots accounts for this brown area, and the small number of the paucity of large dots accounts for the very small dual expansion, clonal expansion here. And then if you look in the tissue and see how this was distributed in each of the tissues, you can see that, and again, here's these three categories correspond to these three categories here, the independent, non-expanded, and expanded. You can see that the most striking is that the cells that were in from the dual expanded clones were also most heavily present in these peripherally clonal expanded ones, and they're smaller when you didn't have peripheral expansion. Again, supporting our hypothesis that when you have expansion in periphery, you have a high infiltration of these dual expanded clones in the tissue. And if you look at the other way, starting from the tissue and saying, can we see what's happening in the blood? We did the converse. We said, if you look at which of these cells were in the dual expanded clones, and then say what percentage of them were in blood, then you can see that when there was expansion in the dual expansion presence in the tissue, there's a high percentage of probability that we could find them in blood. And this suggests that a possible clinical test, that in order to detect the extent to which you have this tissue expansion, infiltration, you could possibly just measure the clones in blood and look for the expansion in blood. So our paper was not the first to do peripheral T cell, single cell RNA-seq and single cell TCR-seq. There were a couple of papers before us that did it, but ours was the largest. But we looked at the previous papers and looked at the same pattern. And we also saw the same pattern. This, again, is a plot. Actually, if I can't remember the, see my, never mind. It's the, so I can go back to the slideshow. Oh, sorry, this is not good. No. Anyway, this is a plot of the expansion in NAT versus in tumor. You can see again this relationship, and then if you look in this other paper again, you see this relationship. Again, when there's expansion, this is expansion in blood versus expansion in tumor. So when there's expansion in blood, and these clones are often expanded in the tumor. And of course, in our study, we did the same thing. We saw this striking pattern across all of our patients that when you saw expansion in the blood, these same clones were also expanded in the tumor. And of course, we have a larger data set, so it's a little clearer. And we did a meta-analysis of all these studies where we measured by one measurement the extent to which you had parallel dual expansion. So this actually was measured by the amount of correlation across to the diagonal for each of the patients. And you measured the extent of peripheral expansion, as we measured in the previous slide. You see this relationship. It's not a linear relationship. It's sort of a non-relationship, but still, when you had a large amount of peripheral expansion, you also saw a lot of parallel dual expansion in the tissue. So there's a relationship between the periphery and the tissue, and it holds across all three studies. So, of course, we now look more closely at the single cell RNA-seq just to understand the different types of T cells that are involved. And we've got this overall clustering. This is called a UMAP plot, where each of the cells is on a position, and they're together and clustered with a different color if they had very similar gene expression profiles. And here we've labeled them by CD8s on the left, CD4s on the right, and you can see these Tregs down here. These other ones that were CD3 had mixtures of both CD8 and CD4. These in particular had a lot of mitochondrial genes suggesting that they were dying T cells. But we looked at each of the clusters, and to show you that the difference between the CD8 and the CD4, as you can see here by CD8, you can see that granzyme K is a good marker of these effector T cells. You can see the CD103 is a good marker of the resident memory T cells, as well as ZNF63, and that there are some differences among these different T cell receptor, these resident memory T cells. And of course, we believe that there are 3 different types of resident memory T cells that we observed here, which differ in their increasing presence in tumor, their increasing expression of this terminal exhaustion, increasing expression of PD-1, and of course, the signature for resident memoriness. We also saw that there was evidence of epigenetic changes going on, and this is a hot topic in cancer immunology. And when we looked at these categories here, this 8.4 chromatin change and this 8.5 mitosis change, we see that that there's a high expression of these histone demethylases and histone methyltransferases, suggesting that these are representative of epigenetic changes. So when we try to integrate now the single-cell RNA-seq and the TCR-seq data, there are certain issues. And one issue is I'm showing the 20 largest clones that we had in our data set from each of the patients. And I've colored them by the different clusters that the T cell came from. And you can see that for the smaller clusters, they tend to often to be somewhat homogeneous. For the larger ones, they seem to be heterogeneous. In other words, every clone can have a mixture of different cells of different types. So that raises an issue. That means that even though we measure the clusters by we look at the issue is that clusters in the RNA-seq are assigned at the cell level, but expansion is assigned at the clonal level. And so to integrate the data, we basically have to say for each clone we can identify a primary cluster. And when we do that, we can see this integration of the data. So here I've shown the single-cell RNA-seq data before on the left, and on the right I'm showing the single-cell TCR-seq data for each cell based on its parent clone. And I'm using this color codes where purple represents a high degree of dual expansion, pink represents a high degree of tumor expansion, and blue represents a high degree of NAT expansion, and then yellow and orange represent these singletons you see here. And what you can see here is that CD4 T cells largely are not expanded. They're largely singletons. But on the other side here, you see a lot of purple. We see that the CD8 T cells are responsible here for a lot of dual expansion. You'll see probably here, we'll get more into that. And also that the CD8 TRMs show a high degree, you can see by the colors here being pink, of this expansion in tumor. And you can see this in a different way by this plot. Here I'm showing, as we saw before, the clone size in the NAT, clone size in the tumor. And you can see that these CD8 T cells have a lot of the dual expanding clones, but there's also this predominance in the resident memory also of this tumor expansion you can see here, especially in the CD8, what we call the 8.3C type. And that you see largely across CD8, CD4s that there's no expansion. They're mostly singletons. And we looked at the other data sets before, we saw a similar pattern. In particular, what we call our 8.1 effector C cells is what these people call the CX3CR1s one, and you see the same pattern, that this particular type of T cell accounts for the dual expansion. And we get to that more clearly when we just look at the CD8 cells. And we look at the CD8 cells and we look at the samples with blood. Previously, I showed only the samples in just the tissue. This now includes the blood, again, with the clone size indicated by the blood expansion indicated by the size of the dot. You can say that really it's this category here. This 8.1 effector T cells accounts for the expansion in blood as well as the dual expansion in tissue. You see some occasional exceptions here, in particular this resident memory T cell, which we're not clear how to make much sense of that. But we do see that majority is due to this effector T cell type. And if you plot these colors back onto the original plots, you can again see that these orange dots lie along the center with the large dots, meaning they're expanded in both blood and expanded in the tissue. And in the patients that didn't have much expansion in blood or in tissue, you don't see this pattern. So we used our data in a different way, not only to look at expansion, but also differentiation. So what we see here is a bar plot, for each of the different types of T cells, and we plotted the number of clones we saw of each type. And you can see here, as shown here by the predominance of gray, that most clones actually in the tissue were not seen in the blood, with some small exceptions shown here. And if we just concentrate and eliminate the gray category and just look at the green and brown category, we can see still that even when a clone had a sibling in blood, shown here in green, most of the time it's just one sibling in blood. Only in a few cases do we see expansion in blood, as shown here in brown. And these were all, as we said before, of the T effector category. But when we looked, and this is in terms of clones, but we looked in terms of cells, though, we saw this interesting pattern. If you look at the cells in tumor, you see that a lot of the cells in tumor, even when they came from these different cell types, these 8.1 cell types, all of these seem to have an expanded clone in blood. And what that suggests is that there was differentiation in the blood into eventually seeing these different types of T cells in the tumor, suggesting that these effector clonal lineages may differentiate into other T cell subtypes. We didn't see this pattern in NAT, interestingly. So when you see the same graph in NAT, you see that, again, there's this infiltration of these 8.1 effector T cells in the NAT, but they don't differentiate into these other T cell types in the NAT. So therefore, in summary, what I'm saying is that infiltration, as measured by dual expansion, occurs into both compartments, but the differentiation is happening only in the tumor. And you can see this here. Again, these are cells in the blood, and you can see there's a high expansion in this effector T cell type. And then if you look at the cells in the tumor, these brown dots seem indicating their parental origin has expanded into these other differentiated CD8 T cell types. So these are all seeming to come from the peripheral blood clones. But we didn't see this in that. So this is a different way of viewing this. So as someone mentioned before, what is the effect of treatment? What is the effect on expansion before and after treatment? And of course, our study only measured pre-treatment samples. But there was a study published recently by a group at Stanford, which did look at samples before and after treatment. And they looked at basal cell carcinoma, which allowed them to sample it after treatment. And what they found is that they found novel clones and tumors. And they also did an analysis which showed there was some overlap, which showed peripheral blood and tumor, but they didn't complete the analysis. And so we took their data and did a more detailed analysis. And what we found was that there was a correlation between the clone size, and because they did bulk RNA-seq, it's not clone size, it's number of transcripts. So they did, I say, bulk TCR-seq. It's a different measurement, but still, this is a proxy for the clone size. And this is the clone size after treatment in the tumor. And you can see this correlation, that when a clone was expanded in the blood before treatment, and then you looked at it again after treatment, they also sent often were these, and these are just the novel clones I'm showing here. So that suggests here. Now you'll notice these different colors, and these are the different colors I'm going to get to in in the next diagram, that we use their classification as cells as being non-exhausted or exhausted. And you can see already the hint of a pattern here, that there's some relationship between exhaustion and the fact that it was not expanded in blood. So, but I'll get to the next slide. But I wanted to look at the variability across patients, because these are only two of the patients, and these are the two patients shown here on the right. If you look at the other patients for which they had data, you actually didn't see a relationship. But at the same time, they just didn't have that many novel clones. And so that suggests that it's the number of novel clones that's determining this behavior of a difference of clonal diversity in the blood. And we looked at the CD4, and we didn't see this relationship between the blood and the tissue. You don't see any dual expansion or triple expansion, really, between what's in the blood and what's in the tumor, and probably the NAT. And so what then said is this could be related to just an overall pattern of clonal diversity. And we measured the amount of clonal diversity. And what we're showing here is we're doing it both ways. We're showing in both in pre-treatment and post-treatment blood. But really, you can see by the number of dots and their distribution along the diagonal that there's more clonal diversity in these patients that had a lot of novel clones in the blood before treatment, and then seeing these same novel clones in the blood, in the tissue after treatment. So it does appear that this increasing clonal diversity is driving the infiltration of novel clones into the tissue. And we can show this over all 6 patients. Of course, there are 6 patients, we're a limited number, so we need more studies. But if you do a measurement of the clonal diversity and the number of novel cells and novel clones, you do see this relationship. So in other words, it does appear that clonal diversity in the blood is what's driving the presence of novel clones in the tissue. To show the relationship between this and exhaustion, we found this interesting pattern. So here again, I'm using this color code as I mentioned before. And the key thing to mention is look at the purple and red. And look at the difference here between those clones which were exhausted and had a sibling in blood, shown here as blood plus exhausted. And they're relatively few. But those clones that were exhausted and did not have a sibling in blood were much greater in number. And these are highly significant relationships. So these difference between exhaustion and their relationship to blood is very interesting. In other words, as I mentioned below here, blood-associated novel clones were not exhausted, whereas the blood-independent ones were more likely to be exhausted, suggesting that exhaustion is largely going on in the tissue, independent of what's going on in the blood, and that perhaps what the blood is doing, it's supplying these non-exhausted cells to the tissue. And you can see by the predominance in the non-exhausted category that these ones coming in the blood are much greater than the ones ending up here in the tissue that were non-exhausted. We also saw this pattern in our data. This is not from the Nature paper, but this is our subsequent analysis. And if you look to see that the expression of PD1 on the y-axis, and you look at the expression of these cells, the expression is highest and I'm using PD1 because it's a good marker of exhaustion. It's much higher in the clones that were found in the NAT and tumor or NAT and tumor both, and did not have a parent presence in the blood. In fact, if the clone also had a sibling in blood, it was much less likely to be expressing PD1. Again, supporting this idea that it's the peripheral T cells that are supplying fresh, non-exhausted cells, clones to the tissue. So this is a cartoon of what we think is going on. So we think that there is 2 types of clonal expansion going on. There's a local blood-independent expansion shown here, down here in this blue, in this purple, of expansion in the tumor only, and these are becoming exhausted. And at the same time, showing here in these yellow clones that there's expansion of blood, it's supplying both the NAT and the tumor, and these are largely non-exhausted. And again, this idea that the peripheral expansion is supplying fresh, non-exhausted T cells to the clones, to the tissue. And then this whole idea forces us to consider whether or not this idea that reversing exhaustion is what's going on. It could be actually the more important thing is this influx of these yellow clones, and that's what's driving the response to patient immunotherapy, not the reversals of exhaustion of these blue clones. And this idea is supported by other findings in the literature, which suggest here that these cells are being epigenetically altering the tox gene, and these are becoming exhausted, shown here. And this, because it's epigenetic, is not reversible. So it might not be that reversal of exhaustion is what's important. It might be the influx of novel clones that are important. And in addition, our also model explains these other findings in the literature. And one finding is that why is it that most of the T cells in the tumor are actually not reactive against tumor? They seem to be reactive against virus. And Simone showed this, that these bystander T cells were reacting to non-tumor antigens. And that another paper showed that only 10% of the T cells in the tissue were actually reactive to the tumor. And our model basically explained this because the influx of cells from the periphery is independent of whether they're reactive to tumor or reactive to virus. It's just simply an influx from the periphery. And to study this further, we did look to see if any of our T cell receptors were found in a database of TCRs known to be reactive against virus. And what we saw for each patient is that for every patient, there was 1 or 2 clones that did seem to be reactive to virus. You can see here in lung 2, this large expansion of green is 1 clone that was reactive to virus. And in lung 5, this one was large, was a reactive effector T cell reactive against the M1 antigen in influenza. So, and again, we supported this by looking at our data collectively, and we saw a hint that the dual expanded clones here, shown here, were more likely to match the viral database than the other types. Again, supporting this idea that what is going on is that you get an influx into the tissue of all effector, these dual expanded, these peripherally expanded T cells, regardless of whether they are reactive to virus or tumor. And of course, the patient could have some viral reaction going on elsewhere, accounting for these viral reactive TCRs. So I'm going to turn now to the clinical implications of our study. And so we saw that different patients have different degrees of peripheral expansion and also this dual expansion in tissue. And the question is, how does that relate to clinical response? And unfortunately, none of our clinical trials in our company had any TCR measurements in either tumor or blood. We did have bulk RNA-seq measurements. And so we're going to try to use the gene expression as a proxy for dual expansion by using gene signatures. We looked at 3 studies, one for bladder carcinoma, one for non-small cell lung carcinoma, and one with in renal cell carcinoma, and they differed. This was only a single arm. This was a dual arm of atezolizumab versus chemotherapy, and this one was a trial of atezolizumab versus a VEGF inhibitor, and then also a tyrosine kinase inhibitor. So, but again, we're interested in the atezolizumab arms as a checkpoint inhibition therapy. And as I said before, since we didn't have any TCR measurements, we had to use a proxy. And our proxy was to develop gene signatures of what clonal expansion looked like in tumors. And we found these set of genes which seemed to correlate with dual expansion, this set of genes with tumor expansion, then this set of genes with singletons, largely ribosomal expression. And you can see here by the increasing fold change along this diagonal that there was a reasonable correlation with the different signature types. Most often, the dual expansion signature was the strongest of these. But in any case, this is our best measurement. And what we did then is we looked to see in each arm of the clinical trial, we looked to see the hazard ratio. So a hazard ratio in each of these dots corresponds to a different gene now. So we look to see the association of each of these genes with the hazard, with response to therapy, and then each signature had because each signature, because we tried to eliminate we look highly correlated genes. This gene signature had 27 genes. This gene signature had 19 genes, and this gene signature dual expanded had 24 genes. And the genes all showed very strong correlation in these arms of the atezolizumab, that each of the genes seemed to correlate with response to cancer immunotherapy. And I'm showing here the ones that were statistically significant in red. And you can see this that in the Poplar study, almost all the genes that represented dual expansion, also tumor singletons, but here dual expansion and tumor multiplex correlated response. And then with the iMotion study, again, we see this this correlation that the dual expanded clones seem to be dual expanded genes in the gene signature correlate with clinical response. So one interesting thing about using these genes, and I'll point this out, is that some of these genes correlate highly with CD8. In fact, I think CD8B is one of the genes here. And so that raises the issue. Is it merely that you're measuring the number of T cells in your sample, because the number of CD8 T cells would be measured by the CD8A expression or CD8B expression? And in fact, you can see this correlation on the left, that although tumor singleton gene signature doesn't correlate with CD8 expression. The tumor multiplet signature correlates highly with CD8A, and the dual expansion gene signature correlates highly with CD8A. And in fact, we also used just a single gene, CCL5, just as a single marker, showing that, yeah, a single marker is also highly correlated with CD8A. And you can see that we already know that CD8A expression already is a good predictor of clinical response. So our question was, well, if CD8A expression is already good, does our gene signature do better than CD8A, or does it contribute an additional piece of information beyond CD8A? And to do that, we did a dual variant analysis of survival. We looked at the patients according to whether or not they were both low in CD8A expression and low in the expansion signature, or they were both high in both CD8A and high in the genes in the dual expansion signature. And of course, you can see by the number of patients, 39 were in this first category, 42 were in this lattice category. They're highly concordant, as we saw before. But there were a few patients which were discordant. There were 4 patients which we had low CD8A and high levels of this dual expansion. And there were 3 patients that had high CD8A but low levels of dual expansion. And so that gave us this interesting parallel, which we saw that only this last cohort category, the patients that were high in both CD8A, indicating the number of T cells, and high in dual expansion, indicating the amount of peripheral and dual expansion, were responsive to atezolizumab. At least this is in the iMotion 150. I'll show you all data later. And these other discordant patients were not responsive. So if you look at all the studies, and here I'm showing the hazard ratio view of this, and what we're interested in are the looking at the concordant patients shown with the filled-in circles, and the discordant patients shown here with the open circles. And then again, we're going to color code them red if they're high statistically significant. And what you can see in almost every case in the atezolizumab arm, the filled-in circle is better than the open circle. In other words, if you were concordant, you had both high CD8 levels and high signature levels, you had a lower hazard ratio, better response than if you were discordant. So what that suggests is that you need both presence of CD8 in the tissue and some indicator of peripheral T expansion for optimal response to cancer immunotherapy. Now, we mentioned before there were 2 types of expansion that we saw in the tissue: dual expansion and tumor-only expansion, this being most of the effector T cells and this being the resident memory T cells. And we then did this dual variant analysis of both. And there was a hint shown here by by these studies where only the bottom category was in red, that was highly significant, that you really needed both dual expansion and tumor multiplet expansion to show the best response to atezolizumab. So in other words, it suggests that the greatest response to cancer immunotherapy happens when a patient has both expansion in the tumor and expansion in the peripheral blood. So since our paper is published, this relationship between peripheral T cells and clinical response has been shown by these other papers here. And these also have monitored peripheral T cell expansion after treatment, and they show basically what we are finding, that the extent of peripheral T cell expansion does seem to be related to clinical response. So I'm going to summarize with the 2 main findings of our paper, or the 2 main ideas in our paper, and then I'll take questions. So the finding 1 is that, again, this importance of T cell expansion and its relationship to clinical response. And our interpretation is that different patients have different degrees of peripheral T cell expansion, representing what Ira Mellman has called the set point of your response. And we think that this really represents your ability to recognize tumor antigens. As we indicated in one of the first slides, the first step in the immunity cycle is to bring antigens to the lymph nodes, and then for the recognition of those antigens to activate and expand the T cells in the periphery. So what we think here is really what seems in retrospect to be common sense, that you really need to recognize the tumor antigens before you can attack the tumor. Without that recognition, it doesn't matter if you give a checkpoint inhibition therapy, since you don't have any effector T cells that can kill the T cell, the tumor cells of interest. So that's this idea that really that we really need both recognition and the cytotoxicity for this effector, for this checkpoint inhibition therapy to work. It also suggests that possibly we could use this relationship between the peripheral blood and expansion, this dual expansion in the tissue, as a type of clinical test, that by simply measuring the degree and variability of the T cell clonal expansion in the blood, you can tell what's going on inside the tissue. And so this suggests a liquid biopsy or a blood measurement, which is easier than getting tissue. And we could use this for both pre-treatment to predict whether a patient might respond to checkpoint inhibition therapy, or we could possibly using it during treatment to monitor the effectiveness of that therapy. Of course, this is just simply an open hypothesis. It remains to be seen whether or not such a clinical tool could actually work. And our second main finding is this finding, and it's the relationship between the expansion and the exhaustion, and this idea that clinical response really is dependent upon the patient's continual response to the tumor. That, in other words, the patient needs to be continually progressing new types of novel clones to be optimally effective against the tumor. And this idea is supported by our finding that there appear to be two types of T cell expansion involving different clones: the expansion of effector T cells in the periphery, the expansion of resident memory T cells in the tumor tissue, and that the exhaustion appears to be associated only with this tumor specific expansion. We showed by the differentiation analysis that these CD8 T cells in the tumor seem to be coming from the expanded effector T cells in periphery. That indicates the direction of the infiltration, that the tissue, that the different T cells in the tumor are coming from the periphery. And this analysis of the Stanford paper suggests that there is a difference in exhaustion, suggesting that the exhausted ones are really clones that came from a previous iteration of the patient's immune system, and that the peripheral expanding clones represent the new iteration of clones, and that there's this continual process going on of infiltration, providing novel clones to the tissue, and that these clones eventually becoming exhausted. And then our clinical analysis suggests that you really need both types of clonal expansion to predict response to checkpoint inhibition therapy. And so that really optimally a patient's immune system needs to be generating new clones over time to the tumor. And so this raises a different perspective on the cancer immunology, that it's not really hot versus cold that's the important thing, although that's important. It's really active versus inactive immune systems, which we're indicating is important as well. So with that, I thank you for your attention, and I'm open to any questions.
Okay, that is okay. Dr. Wu?
Yes.
Can you hear me? Oh, thank you very much.